that brings us to chapter 3, and we're going to talk a little bit about several incredible processes that occur in biology, and these are replication, transcription, and translation. And replication refers to the replication of DNA. So how does DNA make a copy of itself, essentially? We're going to study the biochemical process for that mostly, but we won't really go in that much into detail on that. We will talk about transcription to some extent. Transcription is taking a nucleic acid, a DNA, a DNA strand, and basically copying it and making an RNA strand. And then third, we'll talk about translation. And translation is taking that RNA strand and eventually translating that into a protein sequence, and essentially a protein structure. So we'll first start talking about DNA and RNA structure briefly. So, we've all heard of the double helix. That's what you see in this image here. You basically see two DNA strands that are sort of wrapped around each other to form this helical structure. And what you'll notice here is that you have, each strand along its backbone here, it has these blue things are the phosphate groups. They're shown down here in blue in this structure diagram. And then you have this sort of red link that's there. This is called the deoxyribose group, and this is where DNA gets its name, deoxyribose nucleic acid. And I'll tell you in a second where that comes from. And then the last part of this DNA nucleotide is the thymine base. Actually, there's 4 different types of bases for DNA. Thymine is just one of them. And then the base is this part that's shown here in each case. And, one base comes from one strand, another base comes from another strand. And then they hydrogen bond together, and that's what stabilizes the DNA double helix structure. So what you'll notice is that there's 2 strands of DNA here, and they're just given by these little symbols that are shown here, and these symbols correspond to the bases. So, every nucleotide along the chain has a similar structure in DNA. The only difference is that, each one will have a different base attached to the nucleotide. So in DNA we can have 4 types of bases attached. We can have, it's known as adenine or adenine. You'll see the structure for that in a following slide. T is for thymine, G is for guanine, and C is for cytosine. And essentially these bases pair together, and the bases are what are responsible for the hydrogen bonding. So how this occurs is only thymine and adenine can sort of be complementary to each other, and only cytosine and guanine can be complementary to each other in the DNA strands. So you can see that all of these either have CG or AT across from each other on the complementary strand. What you will also notice is that along this strand, maybe you won't notice it, but the way that the genetic code works is that you essentially have a sequence or a series of bases, and the series of bases is what gives you the so-called genetic code. A codon is every three bases, or every three base pairs here. So, those actually correspond to an amino acid structure, and as you'll see later, this correlates to the ultimate protein structure. But for now, just to at least for this slide, just kind of focus on you have thymine and adenine that are always complementary to each other, and cytosine and guanine, and then the sequence of these bases is what is important. So if we look at RNA structure, RNA is ribonucleic acid, and RNA structure is single-stranded. Okay, and what you'll notice is in this figure, the RNA structure here is meant to be complementary, or as if it were transcribed from this DNA structure. So what happens with RNA is that one of the strands from the DNA, an RNA molecule will be made so that it's complementary to one of the DNA strands. So in this case, this RNA strand is complementary to this top DNA strand. So it essentially has the same sequence as the second DNA strand here, as you can see, but the difference is instead of thymine, it has uracil. And that's because, in RNA, so in DNA, we saw that we had adenine, thymine, guanine, and cytosine. In RNA, we have adenine, uracil, guanine, and cytosine. So uracil replaces thymine when you go to the RNA molecules. So essentially this sequence, it's the same as this DNA sequence on the bottom here. Again, the only difference is that thymine has been replaced by uracil. If we look at the RNA nucleotide, it's essentially very similar to the DNA nucleotide. You have the phosphate group, you have a ribose unit, and this time it's this sugar ribose ring, and it's called ribose in this case. And you can imagine that ribonucleic acid comes from this ribose structure. The main difference here between the DNA and the RNA is, the DNA has the deoxyribose and the RNA has the ribose. The ribose has a hydroxyl group in this position in the bottom corner of the ribose ring. In the DNA, this is gone. So that's where you get the name deoxyribose. And then we have the base portion of the molecule that is shown here. And this is essentially, as I said before, you would have all the bases. The only difference is that RNA does not contain any thymine bases. It replaces thymine with uracil. So these are the actual structures of the bases that are found in DNA and RNA, and these are also found in other important biomolecules such as ATP, ADP, AMP, GDP. There's a host of them, and these are known as the pyrimidine and purine bases. The pyrimidine, and this is just based on their structure. The pyrimidine structures or bases are shown at the top here: cytosine, uracil, and thymine. You can see these are characterized as, you have these almost aromatic rings that contain these nitrogen functionalities in it. You have the amine group coming off at the number 1 position here, and you have a ketone or a carbonyl oxygen here. You can see uracil next to cytosine. It's somewhat similar, except you don't have the amine group shown up here. You have a carbonyl group, and this doesn't have a double bond like cytosine does here. If you go to thymine, You can see thymine has a similar structure also. It has, in addition to, at this position over here, it has a methylene group. Like uracil, it has a carbonyl group up here at the top. It has an amine functionality here, another carbonyl group, and then another NH group. So the difference between uracil and thymine is only this methylene group that's shown here. And then if we look at the purines are guanine and adenine. You can see, their structures are similar. They basically have this complex two-ring type structure that also contains a lot of amine functionality in it. Major difference here is that you can see you have a carbonyl oxygen here at the top in guanine. In adenine you have an amino group. In guanine you have an amino group over here in this position, and essentially you just have a hydrogen on this carbon over here. So, similar structures, but just these little changes that you see in the structures when you go from one to another. This influences the hydrogen bonding capabilities of these molecules, and the hydrogen bonding is what is ultimately responsible for stabilizing the alpha helical structure that we're so familiar with. So DNA structure, this is how oftentimes an alpha helix is sort of represented. You can see inside here, there's different ways to draw, or to illustrate how the bases of the nucleotides interact with one another. You can use these little bars here, or sometimes people actually draw out the molecules to some extent. You can see guanine, it shows that it has like this two-ring structure, cytosine over here with a one-ring structure, and then it's showing like three positions where hydrogen bonding occurs between guanine and cytosine, and then you have thymine and adenine that only have two positions for hydrogen bonding. So it's a nice illustration. You can look at it a little bit differently over here to the right, more to two-dimensional view. So we have, one strand here on the left, one strand on the right. The one on the left, you have this so-called 5' end and 3' end. You can see with DNA, 5' end is matched up with the 3' end of the complementary DNA strand. And then you can see, the phosphate groups, and I guess S must stand for sugar in this case, but it's either the deoxyribose, or it's actually the deoxyribose ring. So you can see these are sort of linked. So this is a biopolymer, and then each one of those deoxyribose rings has, a base attached to it, and then here you can see how the bases from the two different strands interact with each other with these hydrogen bonds that are shown between them. And again, adenine and thymine have two hydrogen bonds. Guanine and cytosine have three. So this stabilizes the structure. This is actually known as the ladder configuration. So this is from your text, and this is one of the things. At the end, I sort of just gave you a few notes on things to read in the text and structures that you should be familiar with. So you should be able to draw a structure like this, showing essentially the deoxyribose or the ribose ring, depending on if we're talking about DNA or RNA. You should be able to draw, the phosphate group on that, and then the base that goes along with that. And again, we're showing thymine and uracil as the bases, but you should be able to draw any of the DNA nucleotide. So you should be able to draw thymine here, adenine, guanine, or cytosine. And then in the case of RNA, you should be able to draw uracil, adenine, guanine, and cytosine. Okay, so how do we go from transcription of DNA molecules to translation, and ultimately, synthesizing proteins in the body. So, as you can see here in this, DNA is our starting material. This is where the information is kept. This is transcribed to RNA and then translated to the protein structure. Essentially, we have, you can see we have the CG and AT complementary base pairs in the DNA. When we transcribe that to the RNA, you can see that the RNA is made to be complementary to the first strand of the DNA. So it pretty much follows the same sequence as the second strand of DNA shown here, with the main exception that instead of thymine, you have uracil in the RNA structure. So once you go from RNA, then that goes through the translation process, and each one of the three, you have sequences of 3 nucleotides in the RNA structure. Each sequence of 3, which is known as a codon, this corresponds to an amino acid. So for example, you would see, you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. You have 12 RNA nucleotides. Since each 3 correspond to an amino acid, you would end up with 4 amino acids that would be, strung together in the form of a protein. So this gives you a little bit of a better picture of transcription, and you essentially start with a DNA molecule, and the DNA molecule, when it is inside the cell, it undergoes this transcription, and you have this enzyme known as RNA polymerase, which is shown here. It essentially opens the DNA structure. When it opens the DNA structure, you essentially have this mRNA structure that becomes synthesized, and it's made to be complementary to one of the DNA strands. So this mRNA, which stands for messenger RNA, is newly synthesized, and then the DNA actually comes back together at the other end of this enzyme known as RNA polymerase. So this is essentially a simplified view of how this works. Another important type of RNA is called transfer RNA or tRNA. So from mRNA, which is messenger RNA, that's actually transcribed to tRNA, which is the tRNA is what ultimately is involved with translation or the protein synthesis. So a tRNA molecule, as you can see here, consists of this sort of, it's known as the hairpin type structure, and you have an amino acid that's attached to it, and then you have you'll have your mRNA molecule. So the mRNA molecule has the sequence of 3 nucleotide bases, and that again is called your codon. And the transfer RNA has what's known as an anticodon in part of its structure. So the anticodon is going to be complementary to the codon. So in this case you can see uracil is shown here. So complementary RNA nucleotides, AAA, will match up with this UUU structure. So you can imagine we have all different types of tRNA structures with all different sequences, and essentially the right one comes in and matches up to each sequence of 3 nucleotides in the mRNA. or for each codon. So this gives you a little bit of a better image of how this works. This is actually a figure that's in your textbook, and I'll give you a reference to that at the end of our lecture. Essentially, you start with the tRNA. You can see that, you can look at the anticodon here, the sequence of the nucleotides that are shown. So you have these different sequences. These, depending on this anticodon sequence, it picks up different amino acids that correspond to this anticodon sequence. And then as needed, there's many different tRNA molecules that are present. As needed, they come down and they match up with the different mRNA codons that are present along this mRNA strand. And essentially what you'll end up with, this all occurs in the organelle of the cell known as the ribosome. You can see the mRNA structure is nicely sort of going through the ribosome structure. The tRNAs come down, and essentially during this process, the amino acids are broken away from the tRNA structures and then formed into a link, which is going to be our polypeptide or protein. And again, the sequence of these amino acids correspond to the codons and anticodons in the mRNA and the tRNA structure. So after, this is showing a little bit further on, after, when you get towards to the end of, making your protein structure eventually, then you'll have the protein fold into its three-dimensional globular form. And then it'll be shipped to the portion of the cell where it carries out its function. So you always hear about the genetic code, and kind of sounds like some mysterious thing. But what we've been looking at so far is what really is the genetic code. If you look at these sequences of nucleotides in mRNA, you'll see that UUU, for example, or UUC correspond to the amino acid phenylalanine. On the other hand, UUA and UUG correspond to leucine. Over here we have UAU and UAC. Those sequences correspond to tyrosine. There are some sequences of nucleotides that are known as like stop codons. So that kind of tells it, okay, you can stop the synthesis of the protein at this point. But this just shows you how you combine the first base, the second base, and the third base to give you the different structures. You don't really need to know like if I have this sequence, I'll have this amino acid. But this is just to give you an idea of how this works. And that's what you really need to know, how this whole procedure works. So we can also talk about DNA mutations, and these are very important. Here we'll look at a normal DNA sequence. So we have the DNA, the two strands, and then the mRNA strand, the complementary mRNA strand, and this sort of TA base pair is shown here as being highlighted. This corresponds to an asparagine amino acid residue, and in any event, this sequence, in this case, the DNA is not mutated and it's in good condition, and you have a normal protein that, let's just say it looks something like this in 3 dimensions. If we have, the same DNA sequences, with one exception where we have what's known as a mutation here. So instead of having TA, we have CG, cytosine, guanine. This would correspond to a guanine mRNA nucleotide. And then, this sequence, now you would have, instead of GAU, you would have GGU. This would correspond to a glycine residue, which is much different in terms of its structure and function than an asparagine residue. And doing so, just changing, the one amino acid, this could have profound effects on the overall protein structure and ultimately the protein function. And you can see here, this is just an illustration, but it shows, how the protein could change its structure compared to the, the correct version over here on the left, just with one amino acid change in the protein. And again, having this different shape would cause the protein to not function in its normal way, because a lot of protein function depends on its 3-dimensional structure, and if one thing's out of place, that'll change everything. So I mentioned DNA replication at the beginning. I'm not, you don't really have to worry too much about that, but you should know that DNA replication is essentially, how we copy our DNA molecules, so our DNA strands. And, cells divide, and, many processes occur in which we need to make copies and pass down the genetic information. And, even like, even in skin cells, you'll have processes that take place, such as mitosis. And in skin, we'll particularly talk about cells of the stratum basal layer, that essentially they're sort of likened to stem cells, which means that they're sort of like the mother cells, and they're pretty much always there, and they divide, and then the daughter cells essentially migrate in the epidermis and become, keratinized cells. But when this process occurs of mitosis or cell division, DNA replication must happen, and essentially what you'll have is, these blue strands here correspond to the original DNA molecule. You sort of have this enzyme helicase, which goes along the 2 chains and opens them up and unzippers them, per se. And when it does that, then you also have the presence of DNA polymerase 3, which is an important, and DNA polymerase 1. And these, are important key enzymes in synthesizing the new strands of DNA. And you can see here, right below this top blue one, you have this sort of red colored one here. This is a complementary, or it's actually the new strand. So the blue one is the parent strand, the red one's the new strand. And then, the same thing on the bottom here, we have the parent strand, and then the new strand that's being synthesized. So, there's a lot of different things that you could learn about this, talking about Okazaki fragments and things, and you've probably heard of these to some extent. But, just the basic principle of, how these things, that this opens up, and then you have, new DNA that's synthesized, that's complementary to the old DNA, and then, the new strands, you'll end up with 2 new DNA helices, each one containing a parent strand and a new strand. So, this chapter was a little bit shorter than the last couple, so we'll end here. But I just want to go over some of the reading assignments in your text that you should be familiar with. So, for right now, you should read Appendix 2, pages 459 to 465. That talks about a lot of the things that we talked about today. Another thing in that appendix, in Appendix 2, there's Figure 1 on page 460 contains the 20 amino acids. You should be familiar with these. You should be able to draw all of the structures of the 20 amino acids. And you should also be able to draw the structures of the DNA and RNA nucleotide. And this was shown in one of the slides here, but it's also shown in Figure 2 on page 461. And then you should also be able to draw, not just the structure of the DNA and RNA nucleotide, but in addition to that, you should be able to draw any of the nucleic acid bases that you would attach on to a DNA or RNA nucleotide. So, these are shown on slide 3. Please note that there is a figure in your text on page 462, figure 3. This is actually incorrect, so no need to worry about that.